Section D.8 is looking at drug action. Essentially, it's looking at how the drug is being designed, how is it going to interact with the body based on its structure. So thus far, we have classified and studied drugs according to their therapeutic effect. And we see that even with underneath stimulants, underneath depressants and analgesics, they are significantly different structures, but they're still cataloged under the same area. So sometimes they like to look at how the mode of action a drug has and classify it that way versus having all identical structures. Because if you look at ethanol versus going back and looking at any of those benzodiazepines, they're both depressants, but they have significantly different chemical structures. Therefore, they belong to the same category due to their drug action. We're going to look at 3 different types, which is called stereoisomerism, the ring strain of the beta-lactam ring, and the solubility and uptake for different drugs. So stereoisomerism, we saw this first in 20.6, but essentially it's looking at the different 3D arrangements within the atoms. Stereoisomerism is the different spatial arrangements of atoms in the molecules. So it's how can you look at them. There are 2 that we're going to look at throughout this. We're going to look at the geometric isomerism. This is your cis and trans setup when you have different groups on a carbon atom. This exists where there's a restricted rotation around the atom, so you have either cis is the same side or trans is opposite sides. Optical isomerism is where chirality exists, and that is where there is an asymmetric carbon atom, so you could get a optical illusion or essentially a mirror image of the molecule. Looking first at the geometric isomerism. Geometric isomerisms tend to be used in anti-cancer drugs. What these drugs do is they go through and they're going to disrupt the DNA function in cancer cells. So to kind of backtrack what cancer cells are, is cancer cells are going to be cells within the body that have gone awry. So our cells have a set number of times in which they can divide, they have a set shape, so on and so forth. And eventually those cells are going to die because they've replicated enough. But cancer cells don't. They don't die, so they accumulate, and they have rapid growth. So that means they have a rapid reproduction or mitosis cycle, and therefore you're going to get a bump or malignancy that there's a chunk of cells that are now what we consider cancerous. So these drugs are going to go through and try to prevent that cell division that's going to occur, that occurs rapidly with cancer cells. So going back to the structure of a DNA, the DNA strand, and it's called the double helix, is connected either side through hydrogen bonds. There's going to be 2 or 3 depending on what the nitrogenous bases are. But overall, the DNA strand does carry a negative charge within the pH of the cell, and we can find DNA within the nucleus. So this is all a review of what DNA is. Okay, so looking at a specific type of anti-cancer drug, there's something called a platinum complex, and this is cisplatin, and I don't know if I'm saying that correctly, but you need to know what the structure of cisplatin is. It is PT NH3 to Cl2. I have seen a question saying what is the structure of cisplatin, so you need to remember that. There's also another version where there's carboplatin that has carbon in them. You don't need to know that structure, just the cisplatin. What they use cisplatin for is testicular and ovarian tumor treatment specifically. And if you look at the way the structures are, they have a square planar geometry. And what this does is it minimizes the repulsive interactions between the electrons in the d orbital. So looking here, we have our cis and we have our trans. What a cis is versus what a trans is if we look at our groups, cis means same side. So our chlorines and our ammonia groups are on the same side of platinum. So remember, this is actually square planar geometry. So we have things coming up and down, forcing our groups into a planar. So this goes towards the extended version of your VSEPR chart. Trans means opposite. So if you look, now your NH3 group is opposite from each other from the Cl group. Again, you have electrons here to create this complex 3D structure. So this is how why we have to use cisplatin versus the trans, and it's the way in which the molecule is set up. All right, so here we have the drug, and we are going to interact this with water. And so what we do is when it interacts with water, you are going to create a ligand, essentially. And it's going to create a positively charged species. So if you look here, we have our Cls and we have our NH3s. So one of our Cls is replaced by water, and the other Cl is there. But you could replace both Cls, as we're going to see over in the next portion. But now we create this positive charge. As mentioned in the previous slide, that our DNA naturally has a negative charge with it. So you can see that the positive and the negative is going to attract. So here we have our reaction with our DNA, and you can see that ligand, the water, essentially carried the cisplatin to the DNA and allowed the platinum group to attach to the guanine on the DNA. Now, like I had mentioned before, you could replace both Cls, and that way you get one platinum attaching to two different Gs or two different guanines on the DNA strand. There are applications for both. It all depends on how far apart the guanines are. So again, this exchanges one or two negative chlorines with a water ligand. This becomes a reactive positively charged species that's going to replace water and the Cl ligands to bind with the guanine base. Now what happens here is usually the DNA is constantly being read by different enzymes within our cells, and they'll go through and fix any kind of mutations that happen during the process, so on and so forth. However, by attaching this platinum or this cisplatin onto it, the DNA repair process is inhibited, which means replication cannot occur, which means the cell cannot divide, and you are stopping the root cause of the cancer cells. But again, only the cis isomer works because it has the proper orientation for the reactions. So the next one we're going to talk about is the optical isomerism. These are what we call chiral drugs. So chiral drugs, a term that we use with them are enantiomers, which forms a mirror image. How this is going to occur is when you have carbon bonded to 4 different groups. So here we have carbon bonded to a hydrogen, a fluorine, a bromine, and a chlorine. And if you were to put like a mirror right down the middle, you create a mirror image. And it all depends on how and which ways the different groups are going to be arranged within the body is going to determine how this drug is going to interact. So what we need to do and go through is when we're going through the whole process of identifying compounds and looking at what we can use for certain drug aspects, we need to look at compounds and we need to identify their identical chemical properties. And so optical isomers can have identical chemical properties. However, One, if you react differently in the presence of chiral bond binding in the body. So only one of the enantiomers is biologically active. And that is going to create the difference in the physiological effects. So if you look at this picture here, again, we have a carbon molecule in the center and it has 4 different groups. And if you look at the molecule in which it's trying to fit in, is the square. This square molecule is going to fit into the square peg, the circle into the circle, the triangle into the triangle. So this drug is going to have the proper orientation. However, if you look at the chiral version of it, so now the square still matches, but the circle will not match into the triangle, and the triangle does not match into the circle. So this one is not correct. So it's not going to give us the physiological properties we are looking for because it's not going to properly react with whatever trigger we're trying to have. So a little bit more about chirality. So we look at the process of biological synthesis, and this means that we are looking at how nature produces these compounds, because most of our drugs come from natural processes, whether they're from plants, from microbes, from yeast, whatever it happens to be. That is usually where we have our starting point, and then we create synthetic compounds from there. So usually within biological organisms, we will produce only one enantiomer form because that is the form that needs to be used for the process in which the body is going to use it. Our bodies do not like to waste, so it's not going to produce 2 enantiomers when it only needs 1. However, when we do a synthetic process, once we have identified what compound we are trying to work with, there are we then go to the synthetic process. However, when we're creating these compounds within a laboratory, we actually will start to get a mixture of enantiomers because we don't really have a good way to control how just the one structure is formed. So the compounds are going to be making all possible structures to make as much as possible. And this is what we call a race mate. And so essentially when we are creating drugs for the body, but in a laboratory, we need to know how many enantiomers are present. And so they call it a race mate because you're going to have not only the compound you need for the disease you're fighting, but you're going to have essentially a mate with it that is not going to help you at all. But before a drug can go onto the market, they must analyze all the physiological effects of each isomer. So if there is any negative side effects, like we had saw with the thalidomide, due to its race mate, we need to know about that because that disaster in the 1960s has changed the way in which we get drugs onto the market. But drugs have to be marketed as a race mate or a single enantiomer so that you know there are going to be potential side effects due to a race mate being present. So if we go back and look at the thalidomide tragedy, it was sold as a race mate mixture. However, at that time, because of how fast we wanted to get it onto the market, they did not complete the research. So if you look at the chemical structures, so here's our carbon, and we have the different groups associated with it. So we have our hydrogen, we've got our group 1, we've got our group 2 and 3, there's all these going on. But if you look at the mirror image, so carbon here, with the way in which the hydrogen is going behind the R1 versus R3 versus R2, this mirror image is what causes the birth defect. So this reacted negatively with the baby. So this had the ability to cross the placenta. This one is the effective version. And they did not do enough research when they were releasing this drug onto the market to know that this chirality would have a negative effect on women. Therefore, we have to go through and look at how much effect can all forms of the same compound have. What is interesting, though, is how this whole process happens. So we have the thalidomide, and we have two forms that interconvert under physiological conditions. So they did not know that that other enantiomer was going to have a negative effect, because it wasn't until it interacted with the biological systems did it actually appear, which is why now we have to go through all those trials. However, they have seen further use for that, the teratogenic, or the birth defect form, which is pretty interesting. So the one that is labeled for the birth defects actually prevents the formation of new blood vessels. So they are trying to see if they can use that version for somehow treating other things, like suppressing tumor growth. Because if we cut off the blood to the tumor theoretically shouldn't grow, so on and so forth. Could we use it in the treatment of HIV and AIDS somehow, or even leprosy? So they're actually, as we build knowledge about these enantiomers, they're finding if the negative ones could go through and actually target diseases and start to go back through the therapeutic process. But to kind of avoid all the problems that could come with race mates, medical fields or the pharmaceuticals are really trying to create single enantiomer drugs more and more often. They take longer, they're a little bit more expensive, but we will know exactly what the physiological side effects are going to be, because it doesn't form an enantiomer. So about 50% of the market today are going to be single enantiomer drugs. All right, the second thing we're going to be looking at is that beta-lactam ring. So if you remember when we were talking about the beta-lactam ring with penicillin, this is what makes penicillin active, is this unique ring that is on the penicillin. So here is that beta-lactam ring again. It creates 4 90-degree angles, which is pretty crazy. But if you look at the way in which this ring is, it looks very similar to two of our amino acids, cysteine and valine, and the way in which they interact with each other creates that ring when they come together. So this is a highly unusual situation to get atoms into, because it forms 90-degree bond angles, which is not very common within nature. Because if we look at the way in which either the cysteine or the valine come in and interact with an sp2 and sp3 hybridization, we would expect the rings to seek an angle of 120 or 109.5 degrees, but it doesn't. It forms 90-degree angles. Well, this weakens the bonds that are going to be made within that beta-lactam ring, and which is going to allow that beta-lactam ring to break easily, and that is the key to the biological activity of penicillin. So that breaking is actually what makes penicillin so incredibly effective. All right, so here is the process when what happens or how penicillin works within the bacterial cell. So something you need to know about bacterial cells is they have a cell wall, but the majority of that cell wall is made of something called peptidoglycan, and it's this network of molecules that spider web to create a really strong structure. Without that peptidoglycan, it becomes like a very traditional cell where we can cause a lot of cell damage due to influx or an influx of molecules, so on and so forth. So this beta-lactam ring reacts with the enzyme, which is transpeptidase, which is used to synthesize the bacterial cell wall. Specifically, it's what's used to make the peptidoglycan in a bacterial cell, which is pretty cool. So what happens is it prevents the polypeptide crosslinks formation. So when the beta-lactam ring goes in, it's going to not allow cross-linkings, that spider webbing of the bacterial cell wall to occur, which weakens that cell wall. And when it weakens that cell wall, we can now that bacteria now becomes much more vulnerable to any kind of changes in its environment. The cell will die at this point and burst, because now it could have an influx of water, it could have a lot of physiological changes occurring, and that is how we're going to go through and kill it. However, we have now seen an increase in bacterial resistance, which we did talk about in an earlier section. I believe it was D8 with antibiotics. So what they have found is why penicillin has become less and less effective over the years since we've been using it, and it's the fact that there are certain bacteria that have an enzyme we call it beta-lactamase, that destroys the antibacterial properties by breaking the beta-lactam ring before that beta-lactam ring can interact with the transpeptidase to prevent the cell wall formation. So it is stopping the penicillin before the penicillin can actually work. So we have created derivatives of penicillin to try to work our way around that. And that is called methicillin, which is what MRSA actually is, which is methicillin-resistant Staphylococcus aureus. So even methicillin now is not as well targeted towards certain bacteria because they are now finding ways in which to break that active ring down. Or oxacillin. But what they have done to create a derivative of penicillin is that on that beta-lactam ring, they've modified the side chains. So by modifying the side chains, that prevents the enzyme, which we called beta-lactamase, and here it could be either term, beta-lactamase or penicillinase. It doesn't matter, but I wanted you to know they call it by the same term. But so you know, ASE means enzyme. So if you remember penicillin, we know we're talking about the drug penicillin, which targets bacteria. Or if you remember beta-lactam, that is the ring specifically being targeted. And so there's two ways in which you can break those words down so you know what it's working for. But because the way the side chains work and they're modified, it prevents that enzyme's ability from going in and binding. So it's going to change the structure. So for those of you who don't know how enzymes work, enzymes have an opening that allows different structures, different compounds to enter. So right now, what we're saying is that this enzyme, if this is penicillin, this enzyme matches penicillin's shape. So it's able to cause penicillin to break down. But what we are doing is we're adding a side chain to it. Well, now by adding this different side chain, this enzyme no longer can react with that penicillin, or now it's called methicillin or oxacillin, and therefore our drug is still effective until new bacteria become resistant to it. The last one we're going to talk about is solubility and uptake of compounds. As we know, polar molecules are water soluble because water is polar. Therefore, if our drugs are polar, they're going to be able to be dissolved in our blood because our blood is an aqueous solution. And it's going to get transported throughout the entire body except the brain because that blood-brain barrier is high in lipids. And therefore, polar molecules do not dissolve in fats, as we know, because those are nonpolar, unless it's extremely small like ethanol. Non-polar molecules, those can cross the hydrophobic blood barrier brain, which is why we modify drugs so that we can hit a target source based on where we need the drug to go, and that will determine how the structure of the drug needs to be done. So therefore, going back to morphine and heroin, as we talked about back in the analgesics, we know morphine has those two OH groups, which means it is polar, but it is small enough that it can cross the blood-brain barrier because it's not extremely polar because it does have some nonpolar sides, but it is still that. So it's going to be less potent. Heroin replaces those two OH groups with ethanoate groups, which decreases the polarity of the compound. That means heroin is able to get through the blood-brain barrier quicker, which makes it more potent, has faster action on the brain, and therefore there's a higher concentration available to the brain, which makes it more potent, which essentially makes it more active. And it's more active by 2 times. And so what they have done is they have noticed that heroin undergoes metabolic changes in the brain. So the esterases, the enzyme esterases hydrolyzes the ester links or the ethanoate links that is on heroin. And so then heroin's actually broken down into morphine when it's in the brain. So this is called a prodrug, where the metabolic products, which is morphine, bring the effects, which is really interesting. So all heroin is it's morphine, but it's taken the polarity away from morphine to increase the solubility into the brain, and then it's just going to be broken back down into the structure of morphine. But because there's more of it in the brain, because it could pass into the brain, we now have a higher concentration, which is really crazy. And so it's just more morphine in our brain than what we could naturally get in there. So this morphine derivative that we are looking at making actually is called 6-acetylmorphine, because acetylcholine is a neurotransmitter that we do use. It only has one ester link, so it's more potent than heroin. It has the ability to get into the brain, but because it only has one ester link, it does not have to undergo hydrolysis and that whole process of hydrolysis once it's in the brain. So it will have an extremely potent effect. And this is produced as a metabolite of heroin. So heroin not only is going to be broken down into morphine, but it can be broken down into this 6-acetylmorphine, which is a very similar structure. And all these different derivatives essentially is what brings on the strength of heroin itself.